Hello everyone, my name is Toshiko Yamazawa of the University in Japan. First of all, I would like to thank the chairperson and committee members for giving me the opportunity to present our research at this exciting symposium journal General Physiology. Today, I will talk about novel type 1 renin receptor inhibitor and novel calcium release via type 1 renin receptor. Excitation contraction coupling is a known physiological function of skeletal muscle. Action potentials propagate along the teachable membrane. On teachable depolarization, DHP receptor mechanically activate renin receptor 1 to calcium release from sarcoplasmic reticulum. The subsequent increase in the cytotoxic calcium concentration triggers muscle contraction. Calcium ATPase suck pumps calcium back into the cell. Here, it is well known RI1 is a key molecule in EC coupling. As you know, hyperactivation of RI1 is involved in several skeletal muscle diseases, such as malignant hyperthermia, MH, and central core disease, CCD. In MH, genetic mutation in RI1 cause hyperactivation of the channel. In CCD, post-translational modification of RI1 may activate the channel to cause calcium leak. Therefore, drugs inhibiting the RI1 channel is useful for these diseases. Dantrolene, the only approved drug for MH. This slide shows structure of RI1. In genetic analysis, more than 300 mutations in RI1 gene have been reported to patients with MH and related diseases. As you can see, there are three main mutation cluster hotspots, which are found in N-terminal region. Region 1 is pink color, central region, region 2, green color, and C-terminal region. Region 3 is blue color. The right panel shows the chromium structure of RI1 tetramer. The top panel shows the structure of the view from the cytoplasmic side, and the bottom panel shows the structure from side views. Red is region 1, green is region 2, blue is region 3. Here is the outline of my presentation. First, novel type 1 renin receptor RI1 inhibitor, oxalic acid derivatives, compound 1. Second, therapeutic effect of compound 1, CPD1, on malignant hyperthermia and heat stroke. Finally, novel calcium release from RI1, heat-induced calcium release. We have characterized various disease-associated RI1 mutants by expressing them in heat cells. Upper panel shows a region 1 mutant. Lower is a region 2 mutant. We found that increased CRCL activity, increased resting calcium concentration, decreased in ER calcium concentration due to leakage from ER calcium store. This is explained by spontaneous calcium leak from ER through the RI1 channel. So this suggests ER calcium content is excellent quantitative index for the RI1 activity. Dantrolene is an existing RI1 inhibitor and approved for treatment of MH. However, dantrolene has a low solubility in physiological saline, so it must be solubilized in water in clinical use. Therefore, development of novel drugs is necessary. Dr. Murayama's group has recently developed novel screening platform for RI inhibitors using ER calcium measurement, and he identified oxalic acid as a selective RI inhibitor with excellent water solubility. However, oxalic acid was less potent than dantrolene. Therefore, they synthesized an oxalic acid derivative and successful in developing a compound 1 with excellent high potency comparable to dantrolene. Does compound 1 have a therapeutic effect? To test therapeutic effect of CPD1, we created mice carrying RI1 mutation R250HC. The mutation exhibits strong MHCCD phenotype when expressed in hex cells. R2509C mouse was generated by CRISPR-Cas9 system. Sequence of an amplified DNA fragment from the of wild type, heterozygous, and homozygous. We found that Homo are lethal, number 1 and 2, but head shows no apparent difference from wild type. This shows PCR analysis of wild type hetero and Homo embryo at 19 days. Is R2509C heterozygous mice are model for malignant hyperthermia? To test the image phenotype of the mice, we exposed mice to ice fullerene and measured Richter temperature. Wild type mice shown no increase in Richter temperature. On the other hand, hetero mice exhibited elevation in Richter temperature and died by fulminant imaged crisis. As you can see, hetero mice responded with full body contraction as reflected in arching of their backs and extension of their legs. We further investigate sex difference in response to isoflurane. There are no significant sex difference in maximum vector temperature, time to death from the start of isoflurane challenge, and from vector temperature of 39 degrees. However, more male mice succumb to isoflurane than females. We tested therapeutic effect of CPD1 on hetero mice. CPD1 was injected by IP, and 10 minutes after, the mice were exposed to isoflurane. 10 mg per kilo CPD1, but not 3 mg, completely inhibit increase in Richter temperature, and all the mice survived. We then tested whether CPD1 can rescue hetero mice from image episode. For this purpose, mice were exposed to isoflurane and at 39 degrees, CPD1 was injected by IP. At 10 mg per kilo, it reduced Richter temperature and rescued all the mice. At 3 mg, Richter temperature once decreased and time to death was greatly prolonged. Thus, CPD1 was rescue effect on MH episodes. It is known that MH model mice show heat score by increased environmental temperature. The hetero mice were anesthetized by IP and put in the test box at 37 degrees. Hetero mice exhibit increased Richter temperature and died. Interestingly, time from Richter temperature of 39 degrees to death was quite similar among all individual mice. Anesthetized mice were placed in test chamber at 35 degrees. CPD1 was administered 10 minutes before a heat stress challenge. Administration of CPD1 delayed the onset of heat stroke, suggesting a protective effect of CPD1 on heat stroke. We next tested whether CPD1 could rescue mice from environmental heat stroke. The hetero mice were incubated in the test chamber and drug was admitted IP. When their Richter temperature reached 39 degrees, All untreated mice died within 30 minutes of time of injection. 10 mg per kilogram CPD1 dose show more pronounced effect. When the drug was administered body temperature of 38 degrees, 10 mmol per kilogram CPD1 dose strongly suppressed the continued rise in the body temperature, and almost all mice survived 60 minutes after its administration. We observed transient effect with the 3 mg per kilogram CPD1 in rescue experiment in both isofuran and heat stress challenge with heteromice. Why is the effect transient at 3 mg per kilogram CPD1? We therefore measure plasma concentration of CPD1 after IP administration. CPD1 rapidly declined in the mass blood flow in first-order kinetics with half-life of about 10 minutes. And it can be metabolized in the liver. In vitro drug metabolism assay using mouse liver microsomes demonstrates that CPD1 reduced to 20% of the original amount at 10 minutes. CPD1 was metabolized more slowly by human liver microsome. In addition, group tests demonstrate that mass strength of wild-type mass was reduced by 15% 10 minutes after administration of 10 mg per kilogram CPD1, and that this defect was almost completely recovered at 60 minutes, suggesting short duration of medical effect in vivo. We tested the effect of repeated application of CPD1 on isofuran challenge of heteromice. As you can see, second and third administration of the 3 mg per kilogram dose when rectal temperature reaches 39 degrees, successfully decreased the rectal temperature. The compound have a therapeutic effect at the other image model mice. Dr. Paul Allen's group created the R163C mice, so we collaborate to investigate the therapeutic effect of CPD1. We examined the effect of CPD1 on heteromice using in vivo measurement of calcium concentration with calcium microelectrode. Resting calcium concentration was higher in heteromice compared to wild type. Administration of CPD1 IP injection effectively reduced calcium concentration in heteromice in a dose-dependent manner. We first tested preventive effect of CPD1. When mice were exposed to isofuran, calcium concentration rapidly increased in untreated R163C mice, indicated MH episode. Pretreatment with CPD1 suppressed increase in calcium concentration in heteromice in a dose-dependent manner, and effectively prevented the MH episode. Also, administration of isofuran had no effect on calcium concentration in wild type. We next tested rescue effect of CPD1. When CPD1 at 10 mg per kilogram was administered to R163C mice 15 minutes after induction of isofuran, it was able to avoid the MH crisis and rapidly decrease calcium concentration to the level measured in wild type. We also tested other MH model mice. Dr. Lopez's group created the G2435R mouse, so we collaborated to investigate the therapeutic effect of CPD1. Home mice were placed in 38-degree test chamber. CPD-1 was administered IP injection 10 minutes before heat challenge. The slope of the temperature increased up to 40 degrees, significantly reduced by CPD-1 administration. Compound 1 was also effective in other MH mice. This is the last part of my talk. How many calcium release mechanisms are there via RI1 channel? The first is deposit-induced calcium release, DICR. Second is calcium-induced calcium release, GICR, which I have already talked about. Third is nitric oxide, NO-induced calcium release, NICR. We found the NICR in the central nervous system. This is the summary of NICR. NICR via RI1 by S-nitrosylation. NICR involved in induced neural cell death. NICR plays a regulatory role in the pathophysiological function of the brain. Fourth is heat-induced calcium release, HICR. As shown in the previous slide, you can see the slope of the temperature change increases with higher electrode temperature. We consider the existence of positive feedback mechanism. We executed in vitro experiment using enzymatic isolated single FDB muscle cells from hetero and wild-type mice. This panel shows immunostaining balance in heavy chain and neuromuscular junction. As you can see, there was no significant difference between wild-type and hetero in morphology. However, at resting calcium concentration was significantly higher hetero cells than in wild-type, and exposure to earth-fluorine increased calcium in hetero, but not wild-type cells. CPD-1 prevented these effects. We then investigated whether there were not heat-induced calcium occur in skeletal muscle cells. When heat pulse of 3 degrees was applied, there were no detectable calcium change during heating in wild type. However, in hetero cells, marked calcium increases were observed during heating. A more strong heat pulse has slightly elevated calcium concentration by wild type, but caused significantly larger calcium burst in hetero cells. These findings suggested that calcium concentration increases in skeletal muscle cells of living animals with the MH mutant in response to only a slight increase in body temperature. I'd like to summarize the main finding of this study. The R2509C hetero mice can be useful as a new MH model. Compound 1 is effectively prevented and rescues MH episodes and environmental heat stroke in mice with 3 MH model. Compound 1 has a great advantage of better water solubility and rapid clearance in vivo over dantrolene. Compound 1 can be promising candidate for clinical useful drugs for RI-related disease. We found that the novel cellular heat sensing mechanism HICR may accelerate the thermogenesis during MH. This study was carried out by many collaborators. I greatly thank these collaborators, especially Takashi Murayama and Nagomi Kurebayashi, for many experiments. Thank you very much for your attention.